The good news is we're starting to figure out stuff. And being in the news, we're starting to see some ways that things work. Also, there are artists that are getting in on the job, because we have, for all the science that comes out, it's published in a journal. And the one good thing about having journals publish stuff is they make it look nice. And they sometimes will have a journal cover. And so there are some journal covers I want to show you from an artist who is doing artistic renditions of antibodies. This is the artist, and apparently she is available. She's even done a cover for somebody's PhD thesis, and I'm like, that would be really cool to have this. If you want to impress your PhD committee, you can contract her to do this. I remember seeing this, and I was wondering, like, who did that? But then I found out that she did other work. It's like, for example, if you look at this right here, this is a replication of the replication of SARS-CoV-2, and you can see the RNA right here that's moving along, and the protein is coming out here. Here's the ribosome. What they have is they actually have a molecule that binds and blocks it from going on, and so it frame shifts, and it's like it moves off the game board in another direction. I love this example of ribosomal frame shifting. It's not literally true, but it makes the whole frame shifting idea. It's literally skipping a base, right? But it literally is going off in another direction when it comes to the information, right? So here's one about oncogenes. When a cell turns cancerous, it basically becomes out of tune. And so here is an out of tune violinist who's turning into malignant cells. But of course, when we have the immune system, it's recognizing, it's chasing after the bad things. Here's SARS-CoV-2. Everyone wants to make it red. I think there's some kind of cultural thing that we just decided SARS-CoV-2 would be red. Probably danger, scary, you know. But then we have the antibodies chasing after it. And I love how these antibodies look, because this is how antibodies work. They have two arms. They're Ys, and their two arms have two hands on them, which are recognition sites for the SARS-CoV-2. I have an association with a certain cartoon. Does anybody else have an association with a certain cartoon when you see this? I don't know if I'm off my rocker here. Looney Tunes? The big, I don't even know what it's called, but it's the big red furry thing. The name of, so you guys see it, right? Okay, so there's a few, maybe I'm not completely wrong about that. I think she took some inspiration from Looney Tunes for what the SARS-CoV-2 eyes look like. And then this is what happens when the antibody catches the SARS, is able to bind, and it's able to bind not just with one hand, but with two. This is an important point for how they work. Oh, and I have to mention the title, You Shall Not Pass. Okay. So what does that's nice to have an artist representation. What does that mean at a biochemical level? Most people can't handle this, but you guys are ready for the truth. Because what we have is we have a protein-protein recognition going on there. And you know that what shows up as a hand-grabbing receptor here is actually two surfaces fitting together, and it's chemistry. Okay? So when you have a protein antigen in yellow, you have the antibody on the right. The antibody recognition site is actually made up of two chains. That's one of the first things to notice. So you notice from this, antibodies must have at least 4 chains. Got 2 per hand. And so you have the recognition site. If you look at that, that's a nice big recognition site with lots of, like 1,000 square angstroms. It's got lots of atoms. This can be very specific and very specifically molded to be the opposite shape to the antigen so that it can bind it exactly. And because we said that is just one part, that's just like this part of the antibody. The rest of the antibody, it has another arm. And if you remember, it has a little body. The body is like the handle of the antibody. And you see that it's different from the 2 recognition sites. So antibodies are really pretty big. You know, those of us who purified fragments, the fragments you designed, they're like 60, 70 kilodaltons, right? Antibodies are twice that big. So antibodies are really big, which means that they're a little bit difficult to make, but they're still just proteins. So it's possible to make antibodies. In a way, it's not possible to like make T cells from scratch, okay? Antibodies also, human antibodies have bound carbohydrate in the middle, so they're not just proteins. So it's not just recombinant technology, although if you look at it, they just have this little area right here. Actually, papaya juice will cleave this. It has a protease, and it's called papain, that will cleave right here. And it will cut off these little bits, and these little bits still work. So it's like a planaria. You can cut it apart, and it will still bind. And this is actually a much more reasonable unit to make by protein function. It will still bind, but it doesn't have the constant region, the blue part down here. We've already talked about this a little bit, so this should be a little bit of a reminder. So the antibody is really made of 4 chains. It's actually 12 domains, and all the domains, if you look at that last one, they all kind of look the same. They're all beta sheets. You can hardly see an alpha helix anywhere. They're all just beta sandwiches. So you have 12 beta sandwiches arranged like this, 2 chains of 2 called the light chain, and 2 chains of 4 domains. The heavy chain extends to the constant region. The other thing is that when you compare antibodies, the bottom part is all the same, but the top part is very different. It's variable, so you call it the variable region, and that's the part that has the binding site. Makes perfect sense. The really important consequence of having 2 binding sites is that the antibody has some chemical activity automatically encoded in it. If you have a lot of the antigen around, each hand gets an antigen. You know, and if the antigen is repeating on the surface of the bacterium, guess what? You've now got 2 handholds on the bacterium instead of 1. And it's much easier to grab on. We've actually measured this in the lab. The ability to grab on with 2 hands rather than 1 gives you another 10 to 100 times binding in terms of KD. So we actually measured that. We compared the situation where you have 1 hand to having 2. And so, of course, when we talk about antibodies, we're talking about IgG. Oh, yeah.
Sorry, I just have one question. Tell me if you don't know this.
Yeah.
Do you know if the complement system is more effective when you have two bindings instead of just one?
Yeah, it's a really interesting question whether complement could have. If it had a situation like this, it would be just as effective as this. It would have the avidity effect, is what we call it. Because this is just technically this is binding cooperativity. So you know that word from 4361 and all that. Cooperativity, it's coming back. It's everywhere. But the whole thing about the cooperativity here is it's an automatic thing. If you have 2 binding sites and you have 2 other sides of the binding site, right, then you will have cooperativity of some sort if they can sterically reach each other. The only thing I don't know is how often it would show up in the case of complement. But I think complement has some things that are multivalent binding proteins. And so I think complement uses the avidity effect exactly like you're saying. Yeah. I don't know nearly as much about complement, although it's really cool because it's entirely protein, no cells involved. So IgG is the common antibody. That's the one that we usually think of, and it's the blue one on the left. There's a bunch of other ones, and actually if you look at the table, you can see it has a long half-life in the serum, 21 days. It has a high concentration in the serum. So it is the blood's antibody. But there are other things. For example, I've worked in the lab with IgA. And IgA has double the molecular weight because it's a dimer of antibodies. And it has different constant chains. But so it might have a different arrangement. But the overall look is it still looks like a Y. It still has 2 hands, 1 feet. But sometimes you can be joined together at the feet. Like IgM here, you have 5 of these, and you've got to be talking about avidity effect is definitely going on here. And D and E are just found in different places. And this is all for the blood. You have immunity in other places as well. Basically, wherever you have an interface with the outside world, you're going to have some kind of immunity. It's not all blood-borne, but of course, blood is the easiest thing to study, and it's the one we know most about. So for example, if you have an allergic reaction, sometimes you have a person who has an allergic reaction to peanuts will have a gut reaction. They'll have hives on their skin. But what's going on is the allergen is getting from their blood to activate mast cells binding IgE. So the IgE is important for peanut allergies. So you just got to look at these different things. And IgE, it's one of the ones that looks like a regular antibody, but it's going to have a different constant region. You see right here, the constant region is attached to the mast cell membrane in this case. IgG won't do this. It has a different constant region. So, of course, you know, just because something has the same secondary structure doesn't mean it has the same primary structure, doesn't present the same surface. And the antibodies use this all over the place. So here's our friend the complement right here. Antibodies can actually activate the complement. There are some components that are just protein-based in the blood, and they're actually probably the first immune system to evolve. I believe we can see complement in many old organisms. But the complement is this little square down here that can bind the antibody. It binds the antibody FC region, and so it has learned how to work well with this other branch of immunity. Antibodies just work by themselves. Let's say that you have a toxin in the blood. They will neutralize it. This is how they were working back when they noticed that people could stand the toxin because they had developed antibodies to it that would bind, and because they bind 2 for 1, they basically, there's no way that this toxin is going to get into the cell because it's all taken up outside. Another thing antibodies can do, macrophages have receptors for antibodies. Because antibody binds a bacterium, it can't completely neutralize it just by blocking it. So the macrophage says, oh, hey, thanks, guys, and it pulls them in. It binds the FC region, pulls them in, and then slowly turns on the acid. It is like the garbage compactor scene. There's another Star Wars reference, so this is the original Star Wars, the garbage compactor scene where they sort of pour stuff in. And that is like they were in the endosome, but that's more cell biology than you really need. So if you look at all these, each of these has an end game. The toxin can be ingested by the macrophage and then recognized, taught to the rest of the immune system. Same thing for all the antibodies. But you realize, and the other thing is that there are some functions of complement that just poke holes in membranes. And so this is working, notice that this is working before it even gets into the cell. This is working automatically just by the protein. Fascinating system, and I have never worked with it, but maybe someday. So here's here's another artist, David Goodsell. Oh yeah, yeah, go ahead. Yeah. Yes, but the question is, can it get to their infective conformation and things like that? But I believe I've even seen it though. The problem with those is they withstand incredibly high temperatures, and part of the problem is that you can't sterilize them off. So there's some other weird physical natures to those that makes the protein itself infectious. If it is non-self, you probably can make I don't know of any non-self molecule that you categorically cannot make antibodies to. And definitely if you're in a lab, you can design it. You don't have to keep the organism alive while you're making the antibody. You can actually design things better. So yeah, you can see a lot of these examples right here. So the bacterium, here's the bacterium in green, and the immune system's basically in pink. So if you look at the different IGs, do you remember how we said that there was IgM, which was like 5 antibodies stuck together, and then there's IgG. So you can see some IgGs here in pink. You see there's a lot of those. Do you see the IgMs? Anybody find the IgM? It's kind of like a hidden pictures type thing. Sort of nod when you see it. Do you see it? There's one here, there's one here, there's one there. When you see the 5 of them together with 10 binding sites, that's an IgM.
Is it not something for complement?
This thing right here?
No,
Oh.
the one that's bound to that looks like
The red one? Which color?
It's pink.
Okay.
The idea, but it's kind of in shape.
Like this?
Just talking about next to the pore.
Oh, next to the pore. Oh, the pore itself might be complement, actually. The pore itself could be dark pink complement. So you think that...
It looks like a bouquet of flowers.
Oh, this right here?
Yeah, that one.
Okay, I think that's an IgM. I think it's just another angle of an IgM, sort of going down and binding a bunch of stuff. But I think this is complement. So I think he might have used dark pink for complement, and I think the red might be like fibrin or something like that, you know. Basically, it's crowded, but if you have enough antibodies, and all these antibodies are going to have the same binding site if they come from the same B cell. So the way that antibodies learn is usually what you have, even with millions of antibodies to choose from. it's still not, it still could be better, right? You know, you look at it and say, could be better. And so what it does is once you have a loose binding fit for an antibody originally in the lymph node, you actually undergo a gene shuffling. And this is why I say it's sort of directed evolution. I want you to see how this looks like Delano and Wells even. They have whole gene segments that they then shuffle. The cell will shuffle, and it'll like put 4 of them together and make a receptor out of 4 of them. And it will even put them together in a messy fashion so that you'll have mutations where they join together. So you have even more variation there. So what it does is, this is how you originally have the different cells, but it can also do some more mutation. It goes into a state of hypermutation, where it has this cell, like let's say this one gets activated, then it will actually hypermutate to try to see if it can get slightly better. So it's a slight misnomer to say they're all clones, but they are very closely related. They're mutants. Yeah. Yeah.
We actually see that the variable is more specific to 3 groups.
Right.
So the actual structure is given by the beta sheets, but then the binding region is these kind of like variable groups, kind of like what we saw when we were designing the practice. Right? We wanted to pick variable groups.
Because you can basically vary a loop infinitely. Not quite, but without losing structure. The loop is not contributing anything to structure. So it's the very tips of these right here, where you have so much variation, you basically have almost random sequences. So this is randomness in big chunks, and these are the loops that you're just talking about. If you take a bunch of antibody variable regions and you plot out their mutations, you see that there's these hot spots. The red places have more mutations in them. Well, you plot that out on the secondary structure, and you see they're all in the loops. You plot that out on the tertiary structure, you see the loops are all together at the end. Boy, it looks like a binding site. And that's exactly what the binding site is. It is hypermutated. The rest of the protein changes a little. But you don't want it to change too much. You still need it to be a beta sandwich, right? You still, what you care about is making the hypervariable regions mutate. These are called CDRs, complementarity determining regions. But I like HV because hypervariable contains exactly what they are. And notice also, of course, this is just one domain. There's 2 domains per binding site. It's a dimer, so it's going to be the same thing in the other hand. So this is fascinating. This actually is a form of evolution. And if you've looked ahead to presentation 4, I'm setting you free again to go out and find me papers about whether evolution is predictable or not. The good news is the answer can be yes or no, depending on how you're looking at it. And here's one case, and I will take any form of evolution, including the evolution of antibodies on the small scale within a body, as well as evolution of species on a large scale. So check this out. There's something here about intrinsic mutability of a particular gene that helps the antibodies develop these correct mutations that will increase affinity. So if somebody wants to take this and sign up for it, just make sure that you sign up for it by name and so nobody else gets to it first. This is the type of paper. This is probably the extreme end of the type of paper that I would accept. And the other end of this extreme is the kind of papers referenced in A World from Dust, especially in chapter 12. So as you're thinking, just keep your eyes open for things that are about evolution and predictability. Or not, right? So structurally, what you're talking about is that you have 3 hypervariable loops that come together at each binding site, 2 chains, heavy chain, light chain, each contributes 3 loops, so you have 6 loops. This is the end-on view, where you have, there's this place in Florida called Gatorland. And to get in there, they have a big gator mouth with the doors inside the gator mouth. You have to walk into the gator mouth, literally like the size of this room, to go to Gatorland. And I don't know if it's still standing, but it's old Florida, predates Disney, really authentic. It's kind of weird to say that a fake gator mouth is authentic, but that's Florida for you. But this is like the Gatorland entrance, okay? It's like this is the last thing an antigen sees before the antibody gets it. And it sees those 6 loops, which are hypervariable. If you have a small antigen, the antibody loops can make a little divot, like a golf hole or something like that. If you have an elongated antigen, they can be like a hot dog bun for the hot dog. Or if you have a hamburger-shaped antigen, they can be like a hamburger bun. You can basically make the surface that will be complementary to any other surface that you have. And if you imagine antibodies against the little tattoo molecules, right? They're going to look something like this. The antibodies against SARS-CoV-2 receptor, they're going to look something like that. So those are the three sizes. This is actually an HIV peptide, so you can have antibodies against peptides. And this is the whole protein. Usually you have antibodies against the intact protein, but you can have them against anything. And so what I really love is when you can go down into the structure of the antibody recognition, and you can say, oh, look, this is phosphorylcholine. This is a small molecule bound to an antibody. You see it right at the binding site. Some loops can't even get in on the fun. It's only a couple of loops here because it's such a small molecule. But here you see the phosphorylcholine, and here you see the phosphate. Here you see arginine right next to the phosphate. Why does this make sense? If you have a phosphate, why do you need an arginine? Yeah, you have a charge-charge interaction. You also have some hydrogen bonds, and tyrosine is right here. I include this, I especially like this one too, because you see tyrosines all the time in these loops. And I even have a paper somewhat about this. It's for another immune molecule. But we found that that molecule's favorite amino acid is tyrosine. And CDR loops, their favorite amino acid is also tyrosine. If you want to know about why, you can read my paper. So the loops can move, but remember how we're not sure if the loops move or not until we check. For example, here's a case where the loops move. If you get a structure of the antibody with no antigen bound, it looks like that. The antigen bound, it looks like that. 3 guesses as to which of the 3 we had small molecule, peptide, and protein. Which of the 3 do you think this antibody binds? Yeah, it's got the elongated thing. Oh, there it is. So it's basically a peptide binding antibody. And so this is definitely a case of induced fit. But remember that I have another paper about this for another molecule. But we had an argument when I was a postdoc with another scientist about whether binding interaction in the immune system was based on induced fit or not. And he said it was because we see the loops moving. And we're like, yeah, they're moving, but that doesn't mean they're important. And we won. So long story short, 3 papers later, okay, but it was really good for my CV because I ended up getting papers. I like it when people fight, because scientists get papers out of it. So the thing is, you can look at these recognition sites, you can ask how they work. For example, you can break them down and say how many H bonds are there. Here's a protein antigen with a lot of H bonds. I love how David Goodsell, again, draws this. He actually has a program right now that will actually apparently do this for you. It will be like PyMol, but it will be for his style. And I've always wanted to try it. I even have an autograph book by him. I talked about him so much about 10 years ago. The students got me his book, and they had him sign it. So again, I have all this stuff in my office. David Goodsell. Goodsell, yeah. So the thing about these antibodies is we can take what the body makes, just like with the diphtheria example. We can, because they're bodies, right? They're proteins. We can take them very easily, take them from one person and put them in another. There's a few other wrinkles to it, right? But at least you see it's conceivable. And now we can make them in vats, because they're proteins, using the same techniques that you all have learned, those of you who have taken 4362 with me. And then we can use those as drugs. And in fact, whenever you have MAB at the end of a drug, that means it's an antibody, a monoclonal antibody. And so there's actually a symbol to it. I did not even know this. It always seems like these generic names are always just made up. And these names are definitely made up, okay? But these names actually have a system behind them. And they tell you what target system they have, what the source system is, whether they were sourced from mouse or human, or they're chimeric, which means you took the loops from one species and put it on another species' constant region, things like that. All that is to say, if you might, I hope you don't know someone who is doing this, but we all usually will have some encounter with cancer in our lives, either in us or those we love, and you will probably know someone who is taking a mAb of some sort. Okay. It might be helpful to them for you to say, hey, we can take this apart. We can figure out where this came from, as long as they aren't too freaked out by species barriers. You know, the fact that there's a mouse antibody. Mouse antibodies work fine. They're mammals, you know, but it might freak some people out. Use your discretion. Okay, so here's SARS-CoV-2. Again, everyone makes it red. And antibodies form against the red viral spike proteins. Now, in early 2020, we already had an antibody that bound the SARS-CoV-2 spike protein. SARS-CoV-2 is similar. So because SARS-CoV-2 was preceded by SARS-CoV-1. And if you look at those sequences, yeah, you can very easily see how the alignment works. I think it's 90 to 80%. This might be where they're most different, in fact. They're very similar. And so you can plot the mutations out, and you can say, maybe this antibody we have in the freezer to SARS-CoV will bind SARS-CoV-2. And here's a paper where they actually found that that happened. Okay? So we had SARS-CoV-2 binding antibodies because we had already studied SARS-CoV-1. So the residues that are different in CoV-2 than CoV-1 are shown in purple. There's not that many in the spike. The red is the antibody, and the brown and gray is the spike protein. This is the receptor binding domain. And so you can see that the antibody blocks right here, so it even looks like it gets in the way of the RBD, the receptor binding domain, which means that it could prevent infection in the first place. Here's some more details about it. The main thing I want to say is you can look down to the atoms to see exactly what these different mutations are and whether it makes sense that the antibody could still bind because it's a conservative mutation or whatever. And then they tested it. This is way back. I wish I had the date for this. This is definitely early 2020. And they found that they could use this human monoclonal raised against SARS-CoV-1, and it would work against SARS-CoV-2. And you see the graphs look pretty much the same. You add more antibody, you see the blue line go down. And so we had those very early on. And this is one of the things that when it seemed like this was completely novel, I was able to say, well, I can read amino acid sequences. And I can see it's not completely novel. And we do have some tools we can do against it. My friend actually posted this. She was like, so this earned whatever this guy is, this meme for it, that llamas could hold the key to a coronavirus cure. And yes, it is true. Llamas are camelids. And crystallographers love to use llama antibodies because their antibodies are simpler, more stable, easier to make in the test tube. Here's the actual thing right here. And this shows, again, you can see that they still have a beta sandwich domain. They're just not showing the rest of the antibody. You can see where all the interactions are, where all the hydrogen bonds are, but I just want to say this is biochemistry. And you can even see some things. We haven't talked about this that much. This is a binding assay. Up means more binding, and we'll describe more for those who need it later. But you can see that it binds in vitro, and then they tested it in vivo, and it actually worked. The final thing you can see is you can actually divide the antibodies into classes. And you can say there's 4 types of antibodies once you get enough structures. This was already in October. This was in August 2020. And they had so many structures of antibodies binding spike that they classified them. And they found there were 4 different types. They put all these together and they overlap them. And you can see it's the same secondary structure for all the antibodies. Antibodies always look the same. And the spike will flex up and down, and you can talk about which antibodies, where the antibody binds will affect this flexing differently. This is the important figure for it. There's 4 different places where the antibody can bind to this RBD. This is the RBD right here. There's the RBD right there. And you have all these different antibodies, and they found there were basically 4 types of antibodies in this world. And they would all have different characteristics. And again, you can take this in the lab, and because you know we can do protein design now, right? So we can do protein design on these once you have a structure, just like what you all have done. So that means it is time. Okay, I have done this. We have 20 minutes to talk about the paper. So I'm definitely not going to get done today. I'm going to run